The first speaker is Dr. Lindsey James. Lindsey is an assistant professor at the Shellman School of Pharmacy at the University of North Carolina, Chapel Hill. The James Laboratory integrates expertise in organic chemistry, medicinal chemistry, chem biology, and chromatin biology in order to discover novel epigenetic inhibitors, while applying these inhibitors to develop an improved understanding of chromatin regulation and to better define the role of epigenetic proteins in disease. She's also director of chemical biology within the Center for Integrative Chemical Biology and Drug Discovery at UNC. She obtained her BA in chemistry from Colgate University and a PhD in bioorganic chemistry from University of North Carolina under the guidance of Professor Mercy Waters. She did her postdoctoral training in Stefan Fry's lab at UNC and joined as a faculty of UNC in 2019. Today, she'll be presenting about an epigenetic modulation by protein degradation. And Lindsey, thanks for joining us. And now the floor is yours.
Thank you so much for that kind introduction. So I want to start by thanking the organizers for the invitation to participate in what's really been a fantastic seminar series. It may be one of the few good things that's come out of COVID. And I tune into these as often as I can, and I've so enjoyed hearing from many different leaders in the field. And many of the talks have really inspired me to think about my own science in new ways. So again, I'm just really thrilled to participate and tell you a little bit about the ongoing work in my lab. So the overall area of interest in my lab is chromatin, specifically post-translational modifications of histone proteins that serve as key signals for the regulation of chromatin structure and DNA accessibility and therefore gene expression. So some examples include methylation, acetylation, and phosphorylation. And there are three main classes of proteins or chromatin regulatory factors that intimately work together to regulate these post-translational modifications. There are the writers that install these post-translational modifications, the erasers that enzymatically remove the post-translational modifications, and then the readers that through non-covalent protein-protein interactions recognize the post-translational modifications. And so importantly, the dysregulation of epigenetic regulatory factors has been an emerging hallmark of disease. And so there are examples of disease-associated overexpression, mutation, translocation, or aberrant recruitment of these proteins that are rapidly emerging. And so the way that we think about this problem as medicinal chemists and chemical biologists is through the development of small molecule compounds, which block the function of these proteins when they are misregulated. And so our expertise is specifically in developing small molecules within the family of proteins that recognize or read methylated lysine. So methyl-lysine reader domains, they generally bind monodi or trimethylated lysine through an aromatic cage, which facilitates cation-pi interactions. And they tend to bind specific lysine residue, as shown on the right, and have a preferred methylation state. And although these domains don't have enzymatic function themselves, they are often part of larger proteins that contain catalytic domains, or in some cases, they recruit protein complexes, which are able to alter the post-translational modification landscape through their enzymatic activity. And so we've taken a few different approaches to developing methyl-lysine reader chemical probes. We developed some traditional antagonists that are, your pretty traditional small molecules that bind in more druggable pockets. We've developed peptidomimetic ligands that are more well suited for some of the surface groove interactions between methyl-lysine reader proteins and their substrates, and often lack what's considered a more druggable pocket. We've been working on covalent chemical probes that are able to covalently label residues within the methyl-lysine reader pocket. And most recently, we've been delving into chemical degraders, which will be the focus of my talk today. And while I know the topic of targeted protein degradation has been covered extensively in this series, I figured I would keep the obligatory slide of how this works, just in case there are any newcomers. And so the way targeted protein degradation works is we can generate these bifunctional molecules that get into the cell and bind both an E3 ligase as well as a protein of interest and form a ternary complex. This ternary complex then enables ubiquitination of a lysine residue on the protein of interest, and then that ubiquitin acts as a signal for degradation of the protein by the proteasome. And importantly, the bivalent degrader is then available to repeat this cycle in a catalytic fashion. And so while we, like many others, have jumped on the PROTAC bandwagon, there are a number of reasons that we think methylation readers are really an ideal target class for PROTAC development. And that has to do with the fact that multivalency really drives chromatin recognition. So methylation readers are generally multi-domain proteins, and often going into thinking about probe development, we lack knowledge of the best domain to target in order to elicit the desired biological effect. Due to their multivalent interactions, antagonism of a single reader domain may not be sufficient to phenocopy genetic knockdown results. These methylation readers are often also difficult drug targets using traditional approaches, and so often we can't achieve the high potency or high target occupancy that is required. And then last, these methylation readers often contain a catalytic domain or recruit protein complex, as I mentioned, with enzymatic activity. So we're largely thinking about ways to develop methylation reader targeted degraders as a way to allosterically regulate histone methyltransferase activity. And one example of this that we published not too long ago is with EED targeted degraders. EED is a methylation reader protein and a core component of the well-known repressive complex PRC2, which installs the repressive H3K27 trimethyl mark. And so Frankie, a very talented and former postdoc in the lab, showed that if you degrade EED, we can potently block the enzymatic activity of PRC2. And encouragingly, a back-to-back paper from AstraZeneca revealed similar findings. And so we were really excited to demonstrate both the tractability of the methylation reader target class for targeted protein degradation, and also that targeting a single protein within a larger protein complex can lead to loss of the entire protein complex. And so for the rest of the talk, I'm going to focus on our efforts to target the protein NSD2. And because this is still a work in progress, I unfortunately don't have a fully complete story to share that's all tied up with a bow, but I thought that it would be more interesting to talk a bit about some unpublished work, and hopefully I can entice you to look out for our publications on this in the future. So NSD2, or nuclear receptor binding set domain protein 2, is a methyltransferase that belongs to the NSD family. As you can see up in the top left, it's a large multi-domain protein, which contains both a catalytic set domain as well as several methylation reader domains. And so it's known for its ability to mono and dimethylate the activating H3K36 dimethyl mark, while the multiple reader domains facilitate the installation and propagation of these marks. And so we were interested in this protein for a number of reasons, some of which have to do with its relevance in cancer, specifically multiple myeloma. And MM is the second most common blood cancer. And currently, it's really an unmet medical need, so we lack a reliable curative strategy. And 15 to 20% of multiple myeloma patients carry a translocation between chromosomes 4 and 14 that lead to NSD2 overexpression. And so this overexpression in turn results in upregulation of H3K36 dimethyl and inappropriate activation of oncogenes. And so there are a number of folks such as Jonathan Licht and others who have done an immense amount of work to validate NSD2 as a potential target in multiple myeloma. On the bottom right, you can see a MM xenograph model that revealed that translocation knockout in NSD2 mutant cells fail to grow following IP injection. Multiple myeloma is not the only place that NSD2 is thought to be relevant. For example, Frank Stegmeyer and coworkers show that NSD2 has a gain of function mutant that's strongly enriched in pediatric ALL. It's been implicated as a conserved master regulated in metastatic prostate cancer. And then I'll also note more recently, Orgazani revealed that NSD2 hyperactivity promotes aggressive malignant lung tumor pathogenesis. So we really felt that there were some novel tools that were needed to really better understand NSD2 biology and its role in disease. And so here I'm showing a chemical probe for the NSD2 PWWP domain, which is the N-terminal reader domain that we published earlier this year in collaboration with the Structural Genomics Consortium. And so since this is published, I've skipped over the 3 or 4 years of work that led to this potent chemical probe. And you can see it's about an 80 nanomolar binder of the PWWP domain. And in collaboration with Jinran Ming at the SGC, we solved the co-crystal structure, showing that the compound nicely fills the pocket where the methylated lysine, K36 dimethyl, which is the canonical mark that the PWWP domain binds, it binds in that pocket. And so one finding that came out of this is that we found that NSD2 binding to nucleosomes really is multivalent as expected with a lot of these multi-reader domain proteins. And so our compound is not sufficient to disengage the full-length protein from nucleosomes. And I should mention that all this chemistry was done by very talented postdocs in my lab, Ronan Hanley and Naomi Menta, who are off at C4 Therapeutics and Neurex, respectively. And so we were able to show that the compound potently engages NSD2 in cells in collaboration with David Dilworth, a former postdoc at the SGC, through a nanobret assay. And so the compound is cell permeable and it engages the PWWP domain in cells with an EC50 of about 1 micromolar. And so our next question was to understand that if we antagonize the reading ability of the PWWP1 domain, does this influence the catalytic efficacy of the set domain, which is closer to the C terminus of the protein? And so we looked at global methylation levels, and we're somewhat disappointed, but not overly surprised to see that when you antagonize the PWWP domain, we really see no change in the levels of K36 dimethyl. And so you can probably imagine where this is going. And so with a potent and selective ligand in hand, we decided to pursue protein degradation as a means to more robustly influence the catalytic activity of NSD2. And so Ronan first tried to implement traditional strategies of appending VHL and cereblon ligands to a close analog of UNC 6934, where the terminal pyrimidine was replaced with a phenyl ring just for ease of synthesis. And we were really striking out big time. And so being the creative scientist that he decided to explore some less well-known E3 recruitment strategies. And so he turned to the NN rule or NN pathway of degradation, which is a process by which the half-life of a protein is determined by the N-terminal and sometimes flanking residues. And so these N-terminal destabilizing residues or N-degrons can be revealed through proteolytic cleavage of a protein, which then serves to recruit proteasomal machinery. The UBR family of proteins is one class of E3 ligases, which recognize these N-degron motifs. And specifically, UBR 1, 2, 4, and 5 are known for recognizing positively charged residues, such as arginine, through their negatively charged binding pocket, as shown here on the right, with the crystal structure of the UBR box domain of UBR2 bound to arginine. And so we were inspired by Rao and coworkers who had appended an arginine to a ligand for ERR alpha and showed that they were able to potently degrade ERR alpha and in turn promote growth inhibition in breast cancer cells. And so Ronan went ahead and decided to append arginine to our UNC 6934 analog, and he made a small set of compounds with a variety of linkers. And again, we were really fortunate to be collaborating with Cheryl Aerosmith at the SGC and her fantastic postdoc, David Nee, who tested our compounds by both in-cell Western assay and traditional Western blot. And so we were very excited to see that the compounds were degrading both the long and short isoform by both traditional Western blot methods and an in-cell Western assay as shown on the left. And this was expected as the NSD2 short form of the protein lacks the catalytic set domain, but it contains the PWWP domain where our ligand is binding. Now, from there, Ronan continued his chemistry, and he started making analogs with the hope of, either increasing the efficacy of degradation or potentially increasing the permeability. And he noticed that some of the modifications that he made to the arginine side chain were inconsistent with what we knew about binding of the UBR box domain to arginine, as shown here on the bottom left. And so we were a little bit puzzled by this finding, and David went ahead and he knocked out the UBR proteins in each case and found that there was no effect on compound-mediated degradation with our compound UNC 7753. And so, we showed that this UBR loss does not block compound-mediated degradation, and therefore is probably not the mechanism by which our compounds are inducing degradation. So with quite a bit of chemistry, we ended up with this compound UNC 8153, which uses this novel warhead to recruit what essentially was an unknown E3 ligase to facilitate potent NSD2 degradation. And you can see here this compound binds or has a DC50 of about 300 nanomolar by an in-cell Western assay, where you start to see a little bit of a hook effect, and then we show degradation by traditional Western blot as well. Now, we show that NSD2 degradation is rapid with these compounds, so we lose about 50% of the protein in only 30 minutes. It's selective, as shown by unbiased global proteomics experiments, and is also proteasome dependent. So introduction of MG132 potently blocks NSD2 degradation. We were very encouraged to see that these degraders do reduce global K36 methylation, as shown here on the right. When we lose NSD2 long and short forms, we see a potent reduction of K36 dimethyl after about 6 days. So this, not surprisingly, showed us that these NSD2 targeted degraders result in distinct pharmacology relative to our PWWP1 chemical probes. We teamed up with Jonathan Licht and his postdoc Amin at the University of Florida, and we showed that NSD2 our degraders have an anti-adhesive phenotype, as shown in KMS11T414 translocated cells. And this phenocopies the result that they have with knockdown of NSD2 in this multiple myeloma model. We also worked with Greg Wong at UNC, and we showed that NSD2 degradation in MM1S cells, which contain this NSD2 gain-of-function mutant. NSD2 degradation in these cells leads to a modest anti-proliferative effect. And so from here, we kind of had two main goals as far as moving the work forward. First, we wanted to see if we could develop more effective NSD2 degraders. And then ultimately, we wanted to determine the mechanism of action of our compounds, of UNC 8153 and our related compounds, which at that point really eluded us. So John Tabor, a very talented postdoc currently in the lab, took over from Ronan. And again, I'm summarizing a lot of chemistry in one real figure here. And we were able to develop these second generation degraders that now have DC50 values, sub 50 nanomolar. And so we were really excited by this progression to be able to improve our DC50 values and better evaluate the effects of NSD2 degradation. We see that these potent degraders have a really robust effect on influencing K36 dimethyl levels. And then because K36 dimethyl and K27 trimethyl are mutually exclusive marks, we also see that K27 levels are increasing with loss of K36 methylation. And those results are plotted and quantified over on the right. Now, from here, we focus largely on, trying to come up with a way to better understand the mechanism of action. And so we turned to a bio-ID experiment to better understand the E3 ligases involved in degradation with these compounds. And just to give a quick summary for how this works, we fused NSD2 with a mini turbo or a Bure ligase. Mini turbo is just a supercharged or superactive biotin ligase. And so by incubating with compound or degrader, what we're looking for is biotinylation of the E3 ligase or E3 ligase complex that is recruited by our degraders to NSD2. And then we can enrich those biotinylated proteins with streptavidin beads and evaluate the proteins that get biotinylated in this assay by proteomics. And so in collaboration with David at the SGC, as well as Brian Rout at University of Toronto, we performed these experiments, and we had a number of different proteins that came out of this analysis, which we are currently in the process of analyzing. We saw a number of different proteasome subunits that came up that were enriched in our 8732 treated samples. And I should mention that we also treated with a negative control compound that that does not bind NSD2 as a comparator. And there was one protein that came out that is a known colon E3 receptor protein that we were quite intrigued by. And so David went into the lab and decided to knock out that protein, as well as the adapter protein in the colon E3 ligase that's known to be in complex with that receptor. And we were really pleased to see that in each case, when we lose the colon substrate recognition receptor protein or components of the E3 colon complex, that we block NSD2 degradation, suggesting that these proteins are likely to be involved in the degradation of NSD2 with our compounds. The next mechanistic study that I'll mention before wrapping up is David went ahead and developed a nanoBret assay with NSD2 fused to nanoluke, as well as our candidate recruiter protein, and found that these proteins form a ternary complex in a dose-dependent manner upon incubation with increasing concentrations of 8732. And so we still have a lot of work to do, both to validate this mechanism of action, as well as explore the utility of our optimized NSD2 degraders in relevant disease models. But overall, we're really excited about where this project is going and some of the somewhat serendipitous discoveries that we've made along the way. And so just in summary, I hope I've shown that we've developed a high-quality chemical probe for the PWWP1 domain of NSD2, and we've utilized this chemical probe to develop potent and selective NSD2 degraders. NSD2 degradation leads to loss of K36-dimethyl and on-target phenotypes. And then excitingly, we've identified and characterized a warhead E3 ligase pair that facilitates NSD2 degradation and has not previously been employed in the targeted protein degradation field, and that we are going to continue to explore so we fully understand the mechanism and how this is working. And then overall, I hope I've shown that methylation reader targeted degraders are an effective way to allosterically regulate histone methyltransferase activity, particularly in cases where the set domains have been very challenging to find small molecules that can alter their activity. So from there, I'll wrap up. I hope I've been able to acknowledge some of the key players along the way, both from my lab and collaborators. Postdocs in my lab that really drove this work are John, Ronan, and Naomi. And then I can't thank some of our collaborators enough, particularly Cheryl Arrowsmith and David Nee, and some funding sources, and I will happily answer any questions.